This conference will now be recorded.
Good morning, everybody. It's nice to say hi to everyone this morning, chat a little bit, and see all your friendly faces. It's definitely been at this a while, and I sort of miss the everyone getting together. If Jesse, I don't know if that's you. Can you mute? I'll give you a thumbs up when to start, just in case that's you. That's doing the reverb back as well. Yeah, so good morning. Welcome to our 2020 Fall Eco-Foci Seminar Series. I'm Heather. I co-lead the seminar with Jens Nielsen, who you will see as well in the chat. He'll monitor that throughout the talk today. This seminar is part of NOAA's Eco-Foci biannual seminar series. It is focused on the ecosystems of the North Pacific Ocean, the Bering Sea, and the US Arctic. And the idea is to improve our understanding of ecosystem dynamics and the applications of that understanding to the management of living marine resources. Since October 21st, 1986, the seminar has provided an opportunity for research scientists and practitioners to meet, present, develop their ideas, and provoke conversations on subjects pertaining to fisheries, oceanography, or regional issues in Alaska's marine ecosystems, including the US Arctic. You can visit the Eco-Foci webpage for more information at www.ecofoci.noaa.gov. We do sincerely thank you for joining us today as we continue our all virtual series. Our speaker lineup can be found online via the One NOAA Science Seminar Series and the NOAA PMEL calendar of events. We are here every Wednesday at 10, except for a few occasions like today, through December 16th. Please double check that your microphones are muted, that you are not using video, and during the talk, please feel free to type your questions into the chat. Like I said, Jens and I will both be monitoring that, and we will address all questions at the end with Jesse. So today we have Jesse Lamb. He's a fisheries biologist at NOAA's Alaska's Fisheries Science Center with us at Eco-Foci. Since 1999, Jesse has primarily dedicated his career as a zooplankton taxonomist. He has expressed that his main interest is how climate change affects zooplankton ecology and therefore the upper trophic level supported by the zooplankton community. He joined Eco-Foci in November of 2015 after many years working on the zooplankton community off the Oregon and Washington coasts. At Eco-Foci, he uses his taxonomic expertise to identify both the zooplankton community and the stomach contents of larval fish of the Gulf of Alaska, Bering Sea, and US Arctic. And with that, I'm going to turn this over to Jesse, who will be sharing his work on the contribution of diet to the dramatic reduction of the 2013 year class of age 0 pollock in the western Gulf of Alaska.
Okay. Am I feeding back at all, Heather?
You are not. You're good to go. Sounds great.
Excellent. Okay, thank you very much. So thanks for everyone for coming, especially on a Monday. Today, hold on a moment. I'm going to be talking about diet data. Its part in the dramatic reduction of or at least what could have been its contribution to the reduction of the 2013 year class of walleye pollock. And I'd like to acknowledge my co-author, Dave Kimmel. So just as a brief background of the fisheries part of Eco-Foci in the western Gulf of Alaska, there's been decades of work on the early life history of multiple finfish species, primarily walleye pollock. And we've done studies in abundance, distribution, recruitment, phenology, habitat feeding, so on and so forth, both within the lab and field study. And from all this data and from all this knowledge, we annually contribute to various reports to inform fisheries managers. So specifically in the western Gulf of Alaska, we've had annual surveys since the mid-90s. These became biennial surveys as of 2001, whereas every other year we either sample the Bering or the western Gulf. And in the Western Gulf in the spring, we have ichthyoplankton or larval surveys, and then in the fall we have H0 surveys. And these surveys also include sampling of both zooplankton and hydrography. So specifically in 2013, our larval survey showed a very high abundance of larvae in what is the Shelikof Sea Valley, which is right here, and mostly south of Kodiak Island. And then that was in May. In the fall, in August-September survey, the H0s that we found subsequently were found primarily southwest of the Shumagon Islands, with a very, very high concentration. So if you compare that 2013 H0 juvenile survey in terms of abundance since 2000, in this plot on the right, you'll notice that 2013 was incredibly high in terms of H0 abundance. This was followed by the recruitment that was very, very poor, as you can see down here. And that table is from Dorn 2019. So the question becomes, what happened to the 2013 year class in terms of recruitment? So this is what I'm going to be focused on today. So I'm going to explore the possible mechanisms of those poor returns, focusing on diets. And I'm going to be working from two operating hypotheses. One was that pollock size and condition was related to their prey choices. That is, we would predict that H0 pollock in good condition would select particular prey of higher energetic content, and that spatial differences in H0 pollock abundances were related to prey distribution. That is, areas with higher pollock abundance will show depletion of higher selected prey, subsequently affecting H0 body condition. So the next couple slides are just going to go over some methods. We used an anchovy trawl for our H0 surveys. They were sorted and enumerated after catching. H0 fish are delineated by a standard length of less than 140 millimeters. And up to 100 H0 fish were measured for body length. And then separately, a length stratified sample of 25 individuals of differing sizes were flash frozen for later data analysis. In zooplankton sampling, we use oblique toes of a 60 and 20 centimeter frame bongo. These nets are all equipped with a flow meter to capture volume filtered. And all of our samples after the cruise have been enumerated at the Polish Plankton Sorting Center and then verified by me at a AFSC. So once we get into the lab, our diet fish are processed by being measured, weighed, and then we dissect the stomachs. For this study, we used 791 total fish. All gut contents are identified to the highest taxonomic level, organized. If I can identify life history stage, we do that, and given a level of digestion. And each prey taxa group was enumerated, dried, and weighed separately to the nearest 0.1 milligrams. So in terms of stats of which I'm going to present today, I'm going to show summary statistics by domain, which I'll go through next. And then we modeled fish condition by using length weight residual values after Broder et al. 2004. And in terms of diet analysis, I'm going to be showing the prey-specific index of relative importance, and then spatial abundance of key prey taxa, that is zooplankton. So PSIRI stands for the prey-specific abundance of relative importance. Sorry, prey-specific index of relative importance. So whenever we classify diets and diet data, traditionally, everything is in terms of proportion. So we use a percent number, percent weight, and then a percent frequency of occurrence. And then from that, PSIRI is calculated. So in the next slide, I'm going to go through a schematic just to go into a little more detail of exactly how this is done. So suppose you have sampled and you have 4 fish. Those are being represented by J. And those fish eat certain prey categories, which are represented by I. From this, we calculate what is a diet matrix. And this is done for most diet data that you ever see presented, where, for example, fish I eat ate 100% of these green circles, therefore that value is 1 in the diet matrix, so on and so forth in terms of number. Now, typically you'll have 2 matrices, one for numbers and one for weights. Just for the brevity of this example, I'm just going to show you a numbers example, but just remember that both numbers and weights are done in exactly the same manner. So at the end of this, we calculate the percent number and then have a percent frequency of occurrence of that prey item. Now, and then you can calculate PSIRI. So what makes PSIRI different than traditional calculation values, which was previous to PSIRI, there was just a calculated value known as the index of relative importance, is that PSIRI uses what's known as prey-specific abundance, which is the percent abundance of weight of prey category I averaged only from the stomach samples from which it occurs. Whereas previous values, the IRI values, would average all of these over all the stomachs. Therefore, instead of 100%, this would be 25, it would be 0.25 here. And the reason for that is because when you average over all of the stomachs, frequency of occurrence has a much higher weighted value because discrete absences are included in the calculations. And the other thing to remember about PSIRI is that it's additive in terms of respect to taxonomic levels. This means that the sum of PSIRI values for different species summed together by, say, their genus will equal the sum of their corresponding family. The sum for that family will equal the corresponding value for their order, so on and so forth. So now I'm going to present some results from our 2013 survey. So this is graphs of both abundance and size. So this graph on the left I showed previously, you have a very high abundance of fish southwest of the Shumagon Islands. And then the graph on the right shows the mean weight of fish sampled per stations, with stations with an X indicate abundance of 0 fish. So you have very high abundances of H0 polyp, primarily southwests, and then the smallest fish in terms of weight were found southwest of the Shumagons, with fewer larger fish in the Kodiak region. So for this study, previous studies done within my group have broken down the Western Gulf of Alaska into multiple regions for comparison. And this is what I'm going to do for the rest of the talk today. So for this talk, domain A is going to be in blue, and remember, it's always southwest of the Shumagons. Domain B will be in gray, and domain C is going to be in orange. So if we just look at histograms of the fish length data from our survey, you'll note that domain A fish were maybe slightly smaller than B, with C being larger than both. And then if we plot similar diet data, excuse me, that is my cats, if you can hear me out. A little distracted. This data are box plots of this length data here, and then we have weight data in this plot, and then abundance data in this plot. So similar than what I showed before, we have smaller fish, a higher abundance of very small fish found in the southwest of the Shumagon Islands. And there's one problem with doing that, in that you have to incorporate the effective sample day from all of that data when looking at differences between the domains. So this is a plot of the survey track of DY1308 from where all the data came from, where the cruise track starts from dark blue and ends over here in light blue. This whole survey was 31 days in total. And what you'll notice is that this represents domain A, this is domain B, and this is domain C. This line right here, there was actually a crew change. So after this station was done, they went back into Kodiak, switched crews, then went around and started back up here again. So that's like a lag of maybe 3 to 4 days. Just remember that. So if you plot the exact same length, fish weight, and now I've included the length, weight, residual data and try to get a measure of condition of the fish, you'll notice that as we moved along in our survey days, the fish got larger, both in length and weight, and that condition doesn't seem to be really different at all. So this plot is just a box plot of fish condition broken down by the domains, and you'll notice that there weren't any statistical differences in fish condition suggested by these domains. And the problem is that the relationship between sample date and domain is that they're collinear. Because we sampled them when we did, and when I broke them down the way I did, it's hard to tease apart fish growth versus actual differences in the fish domains in terms of fish size between the domains. So my co-author and I have been working on this issue. It is Dave's thought that we need to apply some sort of universal growth rate to the fish in order to try to tease apart any domain differences. So for right now, this data is incomplete actually. So now I'm going to move on to some diet analysis results. So what you see here are 3 plots of diet composition of H0 pollock by domain by 10 millimeter length bins. So these are domain A fish, these are B, and these are C. And I know, I apologize, it's probably fairly hard to see these prey categories that you see at the bottom. I'll point out some specific ones later. So what we found from these data are that domain A pollock on top consumed a higher percentage of pteropods and larvaceans, which is noted here. The pteropods are in orange and larvaceans are in lavender. And there was a greater percentage of them than B and C. That domain A pollock consumed a lower percentage of large calanoid copepods. Mostly is made up of the copepod Calanus marshalli, which is in purple, noted here. And that pollock and B and C consumed a higher proportion of euphausids in their upper size bins than in domain A, noted here. So green is euphausids, purple is calanoid copepods, and we should just note that for future slides. So in terms of the PSIRI results, so this graph represents pooled sum values of PSIRI results for different taxa groups. And with the domains A, B, and C, by domain, I should say, A, B, and C. And the things to notice here is that if you notice, in terms of domains B and C, the highest PSIRI values are always euphausids and large calanoid copepods, with some showings of others here, but for the most part, there's a big, you could draw a big line over these and you would notice that primarily this is what they were choosing to feed upon. Whereas domain A had a much larger suite of differing taxa groups. So the weight of the choice of eating euphausids or calanoid copepods was much lower in domain A than in the other 2 domains. So if we look at the corresponding zooplankton data, take it from the bongos, which is what I have presented here, these are the abundances of these same specific taxa groups that I had on the previous slide. So we can see what their abundances were when we sampled them per domain. And the things to notice is that domain A had a very high abundance of pteropods, which was almost exponentially different than the other 2 domains. And that the abundances of larvaceans were higher in both domains A and C. And that the abundances of small and large calanoid copepods were actually lower in domain A than they were in B and C. So in terms of my results summary for abundances, the 2013 year class was identified by a high overall H0 abundance followed by poor recruitment. This highest overall abundance was greater, it was 1.6 times larger than the 2005 H0 abundance, 4 times in 2007, and exponentially greater than all the rest of our surveys since 2000. And then the other thing to note is that the spatial concentration for this survey was unique, where most of the time the average abundances were highest in domain B. And for this study, it was the highest in domain A than it's been, not only during this surveys, but in previous surveys where there aren't that many years in foci past where the spatial scale was as large as these more modern surveys, but the next closest domain A percentage being that high was in the late 70s, actually. For size and condition, the size differences between the domains found in the 2013 survey were found to be collinear. So they corresponded to age growth over the month of sampling, which is currently unresolved. And we did not find any spatial differences in fish condition as of yet. For diets, domain A had showed a greater preference for a broader variety of taxa. And then domain B and C had a preference for primarily two groups, euphausids and large calanoid copepods. And lower abundances of small and large copepods in domain A could suggest that they were preyed down upon by H0 pollock. So the question still remains, what happened to 2013 year class recruitments? Why were the returns so low? So over the next couple slides, I'm going to go through some other possible causes for low recruitment in that 2013 year class. One of these could be density dependence, poor diet affecting overwinter survival, cannibalism, and advection. So one at a time. Density dependent. So one could say that high abundances of H0 pollock found in domain A could increase the likelihood of overwinter mortality due to density dependence. The problem with this is that we've had very little evidence of food limitation. You'll note that, even though domain A fish ate many pteropods and larvaceans, they still ate a fair amount of euphausids. And that higher abundances of pteropods and larvaceans in domain A does not indicate competition. That lower abundances of Calanus marshallii could suggest density-dependent competition, but this is impossible to test within our data sets. So there's actually fairly small evidence of any sort of density dependence. Even if there was, we don't have any real way to prove it. So in terms of poor diets, so pollock overwinter survival and recruitment success increases when fish attain a larger body size and condition. And there have been many, many studies that have shown that marine zooplankton accumulate storage lipids, which have a very high energy content. So two of these taxa with very high lipid content are both Calanus marshallii, which is a large calanoid copepod, and species of euphausid. So euphausids and calanoid copepods have a very high energy. So everything that I'm showing you here is represented in kilojoules of dry weight per gram. So euphausids, it's about 21 and 25, yet for pteropods and larvaceans, it's around 12 and 15 kilojoules per dry weight. And the other thing to consider is not only are these high energy prey taxa, they have higher energy, but you have to consider the relative size of these taxa as well. So in terms of comparing just sizes of these animals, euphausids are very large. It would only take you, a couple, 2 to 3, 4 adult euphausids could easily equal 1 gram worth. Copepods, that Calanus marshallii, it's probably, closer to, 15 to 20. But then the amount of larvaceans and pteropods that you would need to equal 1 gram of dry weight is so many more. So that's something to really consider as well. So when you're preying on euphausid, you're getting much more bang for your buck with just eating 1 euphausid than you ever would just 1 larvation or 1 pteropod. So this would suggest that that an increased proportion of lower quality prey consumed by pollock possibly face an increased overwinter mortality. So the next possible cause, cannibalism. So here's a backstory. So I've been writing this in a manuscript now pretty much in the spring and over the summer. And I've sent these, I've sent out the manuscript to be reviewed by, in-house within our group. And at the very end of my discussion of one of my manuscript reviews, Lauren Rogers put in like this one little sentence comment at the very end. She's like, I like all of these reasons, but in the end, they probably just got all ate upon by the 2012 year class. It wasn't offhanded, but it was definitely like, oh, like something that nobody actually ever brought up with me before. So if you look at this, remember this original graph from Doran et al. In 2019, you'll notice that this abundance of age 1, 2012, these are the 2012 age 1 recruitment. You have very, very, very many fish. And if you take this and then you look into a study by Sogard and Ola from 1994, which studied intra-cohort cannibalism in a laboratory setting, they showed a combined 47% mortality of smaller pollock when attacked by larger ones, with 11% of them swallowed whole, and just mortality from being attacked at 36%. So this probably had an influence on returns of those 2013 fish. So the last possible cause I like to go through is actually from an incredibly recent paper by Matt Wilson and Lehman from 2020. And this suggests that evection has an influence on age 0 recruitment in the Western Gulf. So they examine age 0 pollock distributions for evidence of high abundance southwest of the Shumag. That high abundance of a pollock southwest of the Shumagins does not translate into higher abundance at older ages, possibly due to evection. That surface winds can reasonably explain variation in age 0 distribution. And then they examined relationships between surface winds, age 0 distribution, and recruitments. So here are just a very brief overview of some of the results and discussion points. So they found that the highest population density of age 0s occurred in the southwest region and domain. And just to note, this graph right here is the exact same graph that I showed in my previous slide. So it's from the exact same year, it shows the exact same things. And that lower age 0 abundances they found lower age 0 abundances off of Kodiak and Shalikoff regions. But then in terms of predicting recruitment, 96% of variation in pollock recruitment was explained by the age 0 population density. So what that means is that the higher age 0 population density that you found within these regions was informative of predicting when the recruitment of subsequent years, which you'll notice in both, this is age 0s on this end, and this is age 2s on this end. And you'll see it for both here and here in 2015 and 17, you had a higher age 2 returns, whereas in 2013, it wasn't anywhere near as high. And that when they tried to test it from the Samedi in the Southwest, they found no significant relationships at all. So this is the same 2013 plot that I showed previously, just grown up from this slide. So the other thing that they showed was that what they call wind hypothetical displacement, which is a cumulative sum wind vectors in kilometers, was the strongest in 2013 of all of their 5 study years, where during 2013, they had some form of southward displacement. This means that they had the strongest southward, southeastward, or southwestward winds from these 4 NDBC stations found here, here, and here, and that strong wind, they suggest, displaced the age 0 abundances to this region. And that strong southwest evection most likely also influenced the pollock recruitment. So in summary, the recruitment of this year class probably got diminished from a combination of larger portions of the 2013 fish feeding in lower energy taxa. It suggested that cannibalism from the large 2012 year class, which has not been proven, but it's highly suggestive, and that large portions of the 2013 age 0 pollock evected to the southwest. So they were evected in the southwest, and that southwest evection, those fish being there, does not inform recruitment into the next years. So my future work is to publish all of this data. And with that, this is my acknowledgement side, and I will take your questions.
Thank you so much, Jesse. Okay, if you want to put on your video, I will do the same.
Okay.
And hey,
Okay.
all right.
Why isn't?
And your audio, do you need to swap that again?
Yeah, can you hear me?
Yep.
Okay.
And then
Yeah,
do you
I think
want
I'm good now.
to put it back in presenter mode and just put it back on the title slide?
Okay.
Thanks. And Libby says you had a great talk, and Colleen says clap, 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 which I usually do for everybody. So there you go, clapping for everyone. Thank you. Great job, Jesse. And obviously, if you guys have questions, please go ahead and put them in the chat, and we'll monitor through those. And while we're waiting, Jesse, here's the question: is there any way to validate or confirm the cannibalism idea? Do we have?
So the best way to do that, I think, would be for me to contact folks downstairs in Groundfish who do diet work from that year, and actually, confirm with them. And then, yes, I think that it is possible, but I have not done that as of yet, to ask to see if they saw a higher proportion of H0s within their adult pollock stomachs from that year.
But that is data. I mean, that's data we collect not necessarily we as but
I believe so. Yes.
Cool. Peggy says, yes, thank you. Interesting and informative talk. Hi, Peggy. Any questions from folks? Any? Just waiting. So it sounds like you've already written this up.
Yes, it's already actually gone through internal review.
Nice.
So yeah, it's just, I'm waiting to fix that one pesky problem with the fact that the fish grew while we were sampling them. But once I squeeze through that, then we're going to be sending it off.
Congratulations. And if you guys didn't know this is Jesse's first foci seminar talk. So that was a big deal in our group when somebody gets to do their first seminar in there with us. So 5 years. Only took 5 years.
I have a question, Jesse.
Oh, hey,
Yeah.
Jens.
This is Jens. So you talked about the condition of the euphosates and Calanus being much higher than larvaceans and pteropods, right?
Mm-hmm.
Do you know anything about whether condition of, for example, Calanus also varies, and if that might have something to do with it? I'm thinking if they don't accumulate in sort of lipids like they normally do, maybe because of food shortage for the Calanus or along those lines, if that makes sense.
Yeah. Well, it's an excellent question, because this is something that we're going to be working on actually in the future, measuring both lipid content and just relative size of the Calanus from year to year, and how much of an effect that would have on feeding in general. But to go back to, for that year, for the 2013 fish, what we have to think about also is the climate conditions overall in the Western Gulf. So 2014, 15, and 16 were the warm, quote unquote, bad years. Yet 2012 was actually the last really cold year. So it seems as if 2013 is kind of like intermediate. So one would think that in terms of, Calanus being at a higher lipid level, in comparative years, you would think that, colder years are usually better in terms of them getting as fat as possible, shall we say. So I don't think 2013 was the worst year for them. It was kind of like a transition year almost. So I don't think that would have as much of an effect. If this had happened in 2014, 15, and 16, that's an excellent question that would need to be looked into further. But not for this year, I wouldn't think.
All right, thanks.
So Martin Dorn had a question as well, Jesse. He said, any speculation about the role of the 2013 year class in seeding Eastern Bering Sea pollock?
That's all right. So I actually, when I initially did this, and this has been discussed in other papers as well, whether or not that high population that was found in the Southwest was evicted through Unimak Pass and would have done that very thing. And I find it interesting. I tried to figure out whether or not you could get, what sort of measures we would have to get, a slug of that population like through Unimak Pass and just around into the Bering. And I think it's an interesting study, but I remember an interesting thought, but I remember in just our little, reviews within our group that it seemed to be downplayed by other members of my group. And I'm not like, the best person of information on that, although I would love to look into that more.
Martin, I hope that answers your question. You're more than welcome to unmute and follow up if you'd like. Give him a minute. He says, yes, great answer.
Cool.
Jens, do you see any other questions here in the chat?
I do not. And I didn't get any written to me personally either.
Okay.
I can ask one more if that's okay. Just keep Jesse on the spot. So if I remember correctly, and I may or may not, and Martin Dorn can probably correct me, or Lauren, but I seem to recall that the 2018 year class was really high but has sort of disappeared. Do you think, I think I'm correct on that? Do you think similar mechanisms to 2013 occurred, or do you think it's much more in 18, or do you think it's much more related to these really warm 2019 temperatures? Or do you think some of the work that you've done here may also explain some of what's happened lately? That's maybe not a fair question, actually. But.
Yeah, well, okay, so I haven't looked at their diets yet, which I will. But my guess would be it would be much more. Not only do you have like maybe the food was in poor condition because of warmer conditions as well, and just plankton being just lower in that year, but then the other part of that is that you always have to remember that with increased temperatures and the whole thing, that these fish would be growing faster, and metabolisms will be jacked up maybe a little more. And I think what I think that, like, stepping back from, like these questions is that, I mean, and this shouldn't be a surprise, but I don't think it's ever one thing. I think it's always a suite of things, and, and the proportions of what, each one and the other is. Like, from my talk and from what I've done, if I had to guess, I'm guessing that cannibalism and that Southwest eviction were the two biggest factors in the poor returns of the 2013 fish. But then in the more recent years, I would probably just go to climate and just poor feeding conditions. That would be my suggested answer for that.
Great, thank you.
Yep.
Should I jump in, Heather? This is Martin Dorn.
Go ahead.
I don't know how easy it is to have a discussion, but we can give it a try. It's a little different for the 2018 year class. I'm just wrapping up the assessment, so it's right on the top of my mind. But what happened there is that it was very abundant as age ones in the Chillicothe Strait acoustic survey, and then sort of vanished after that. So it was sort of the transition from age 1 stage 2 that is sort of the puzzling situation right now. And I don't know if anyone has a good explanation for that yet. We've been sort of thinking about it. It's very striking in terms of starting off very large and then getting small, but it is different, I think, than the sort of things that we thought about for this study.
Okay.
Thank you, Martin. Does anybody else want to jump in that might have information as well?
It's always kind of awkward doing these kinds of studies because you guys are like every other year sort of in the Gulf, and it's just like we can't have a definitive study about the 2012 year class because we just weren't out there, and it was, you know, and then we're kind of sort of constantly sort of dealing with this, and that we can only get information about what's going on in early life history every other year, and it just doesn't work very well. At any rate.
Yeah, I wholeheartedly agree with that. That's the one thing from doing this study is that what I realized, you know, this is like a couple of years ago, that like, oh, we weren't out there in 2012. I'm like, oh man, like that's, so yeah, it's an excellent point.
And does that change your strategy? I mean, like in your outcome of missing every other year?
It's you just got to work with what you have, really. I mean, it's tough. Even if we were to be able to do like, you know, some sort of smaller something, just to have anything, you know, year to year would be great. But yes, it's brought up more often than not.
I see Bob was on the phone. You can talk to him about that. I'm saying that with a smile, Bob. Let's see. Okay, I don't see any other questions. And I will give folks a minute to do that. And I'll just say thank you, everybody, again for joining us. Thank you, Jesse, for signing up to do the seminar and giving a talk. And just to remind folks, we'll jump back to Wednesdays next week. And Jennifer Provencher will be talking. She's a conservation biologist at the Environment of Climate Change Canada in Ottawa. And she'll be discussing fisheries and seabird bycatch in the Canadian Arctic. So we'll move up a few regions next week. And if you miss a seminar, we do have them recorded. It takes a couple weeks to put them up, but they are on the Pacific Marine Environmental Laboratory, so PMEL's YouTube page, and you can search EcoFoci seminar and they'll be listed there. So Jesse's will be there in a couple weeks once we are able to verify all the verbiage. It doesn't work perfect. So, yep. So Ashley, as I just mentioned, the recordings will be posted. So just touch base with the PMEL YouTube page in a couple weeks, and you should see it up there. And Bob Boyce does great talk, as does Mary Beth and Michelle and everybody. So you can read those as well, Jesse. But again, thank you everybody for joining us. Thank you, Jesse. And
thank you.
And we'll see everybody next week. Okay.
Okay.
See ya.